Meiosis is a process of cell division that, unlike mitosis, halves the number of chromosomes in a parent cell, ultimately creating four daughter cells called gametes that are each genetically distinct from the parent and from each other. Gametes, or sex cells, are different from the diploid parent cells in the process of meiosis because those diploid cells have paired sets of chromosomes, while a haploid cell, like a gamete, has unpaired sets of chromosomes. Thus, the process of meiosis represents a cell division that takes a parent cell that is initially diploid to the state of four genetically distinct haploid cells. This video shows an example of a 2n equals 6 cell, meaning that there are two sets of 3 types of chromosomes, giving a total of 6 chromosomes. Before a cell undergoes meiosis, it must undergo interphase, which, although technically not part of the process of meiosis, is still essential for the cell division to occur. During the G1, or first gap stage of interphase, the cell begins to grow and expand, while also the centrosome duplicates. After the growth of the cell in the G1 phase comes the S phase of interphase, in which the cell duplicates its chromosomes. This S phase consists of DNA synthesis and replication, and results in the formation of two sister chromatids, which are exact copies that are joined together at their centromeres. Next comes the G2 stage of interphase, in which the cell continues growing as it has been growing throughout the process of interphase, all the while preparing itself for the actual division process of meiosis. Following interphase is meiosis 1, in which our initial cell is going to divide into two unique daughter cells, which will both be haploid, meaning they'll have only one set of 3 chromosomes, or half of the original amount of genetic material that we had with our parent cell. The first step of meiosis 1 is prophase 1, and in prophase 1, the centrosomes that previously duplicated in interphase start to move further apart from each other inside the cell, and spindle fibers also start to form. During prophase 1, chromosomes also begin to undergo homologous pairing, where each pair of the same type of chromosome aligns, and each individual member of that pair is called a homolog, and one homolog is from the mother and one is from the father in terms of the set of chromosomes that they're coming from. So in this case, there are 3 homologous pairs of these 6 pairs of sister chromatids. At this stage in prophase 1, the chromosomes also undergo something called crossing over, and in the process of crossing over, 2 homologous chromosomes exchange genetic material, and that exchange of genetic information is what allows for variation within the genetic code of the ensuing daughter cells. As the centrosomes start to move to opposite sides of the cell, the spindle fibers that are trailing from the centrosomes attach to the centromeres of each of the chromosomes. As the centrosomes continue moving to either pole of the cell, the homologous pairs are subsequently aligned in the center of the cell. In the next stage, which is called metaphase 1, the pairs of homologous chromosomes begin to line up at the metaphase plate, which essentially delineates the center of the cell, and it's the line across which the cell is going to divide in later stages. In the next stage, anaphase 1, the homologous chromosome pairs are separated as the spindle fibers from each of the opposite centrosomes of the cell begin to pull at the centromere of the aligned chromosomes. Telophase 1 is the final phase of meiosis 1, and in that phase, the 2 haploid daughter cells that have been forming throughout this process are finally produced. Each of the chromosomes in these 2 daughter cells still has 2 sister chromatids, and simultaneously the process of cytokinesis occurs during telophase 1, which effectively creates a cleavage furrow that is formed between these 2 individual daughter cells as a result of the splitting of the cell's cytoplasm. This concludes meiosis 1, and at this current stage, the individual initial cell has split into 2 genetically distinct haploid daughter cells. Meiosis 2 completes the overall process of meiosis and results in the formation of 4 gametes, which are haploid daughter cells that will result from the splitting of the daughter cells produced in meiosis 1. In prophase 2, a spindle apparatus forms following the duplication of the centrosomes in each of the daughter cells that were formed in meiosis 1. During prophase 2, the centrosomes also begin to migrate to opposite poles of the cell, but unlike in prophase 1, there is no crossing over or homologous pairing of the chromosomes. Again being pulled by the spindle fibers, which are attached to the centromeres of the chromosomes enter the next stage of meiosis 2, which is called metaphase 2. And in metaphase 2, the chromosomes again line up at the metaphase plate of each of the respective cells. The chromosomes in each of these daughter cells still consist of 2 sister chromatids that are joined at their centromeres. In anaphase 2, however, these sister chromatids are separated, becoming 2 individual chromosomes for each original chromosome in the earlier stage. Finally, the process of cytokinesis and the splitting of the cytoplasm during the next stage of meiosis 2, which is called telophase 2, results in the creation of 4 haploid daughter cells. These resultant gametes each have one set of 3 unduplicated chromosomes, and due to the process of crossing over that occurred during prophase 1, each of these gametes is genetically distinct from both one another and the parent that produced each of these 4 gametes. This concludes meiosis 2, and resultantly the entire process of meiosis. Throughout this process, there are many connections to Mendel's 5 claims. Although Mendel's 3rd claim about the dominance of certain alleles and how that relationship occurs in Mendelian inheritance is not actually supported by the process of meiosis, the other 4 claims that Mendel put forward are. Mendel's first claim that alternative versions of inheritance factors are what accounts for variation in the characteristics that offspring inherit from their parents is supported in meiosis by the fact that each of the haploid daughter cells produced is a separate gamete and contains distinct genetic information. Mendel's second claim, which is that an organism inherits 2 copies of an inheritance factor, one from each parent, is shown in meiosis because the resulting cells that are produced as a result of this reduction division of meiosis are in fact gametes, and gametes have only one set of genetic information that, when they combine with the gamete of the opposite sex in the process of fertilization, the somatic cell that is formed contains 2 sets of chromosomes in homologous pairs, one set received from each parent. Mendel's fourth claim that the two alleles for a heritable character segregate during gamete formation and resultantly end up in different gametes, which is also called the law of segregation, is actually represented during anaphase, in which each pair of chromosomes containing two alleles for each gene is split up in the process of anaphase, resulting in a division that requires that each daughter cell only contains one of the original pair of two. Finally, Mendel's fifth claim, which is that two or more genes assort independently, and that each pair of alleles segregates independently of each other pair of alleles during the process of gamete formation, is also represented in meiosis. This law of independent assortment is supported in meiosis 1 in the metaphase stage. Homologous chromosomes end up aligning along the metaphase plate in random sequences, with their distribution and their ordering along that metaphase plate having no relationship to the ordering of other homologous chromosomes. This shows that chromosome pairs and homologous pairs do not have an effect on one another. They distribute into daughter cells randomly and effectively assort independently of one another. Thank you for watching our video on meiosis. We hope you learned a lot.